Hello. This begins the Unit 2, Module 5, Chapter 10 presentation. The big picture topics for this presentation include a discussion of the principles of taxonomy and a discussion of phenotypic characteristics that can be used to identify and distinguish different bacterial species. The system of binomial nomenclature allowed prokaryotic organisms to be named and grouped together. The refinement of the classifications increased as more detailed information about morphology, physiology, and biochemistry emerged. Nucleotide sequence information has revealed very unexpected relationships and deviations from previously established classifications. Let's begin with a definition of taxonomy. Taxonomy encompasses the description, identification, nomenclature, and classification of organisms. The taxonomic categories from top to bottom in this diagram are further subdivided into more refined categories. Thus, organisms at the top of the hierarchy in a kingdom share only one or a few characteristics, whereas members of the lower category or categories share the majority of their characteristics. A species is one of the basic units of biological classification and a taxonomic rank. A species is often defined as a group of organisms capable of interbreeding and producing fertile offspring. Different measures are often used, such as similarity of DNA, morphology, metabolic characteristics, or ecological niche. Phylogeny is the study of the evolutionary relationships among groups of organisms, for example, among different species or populations, which are discovered through molecular sequencing data and morphological data matrices. The result of phylogenetic studies is a hypothesis about the evolutionary history of taxonomic groups. This is referred to as their phylogeny, and it's visualized in a phylogenetic tree, which you can see in the diagram. The tree represents a hypothesis of the order in which evolutionary events are assumed to have occurred. The current three-domain phylogenetic tree is based on the analysis of ribosomal RNA. Carl Woe's elucidation of the tree of life shows the overwhelming diversity of microbial lineages. The single-celled organisms represent the vast majority of the biosphere's genetic, metabolic, and ecologic niche diversity. As microbes are crucial for many biogeochemical cycles on Earth and to the continued function of the biosphere, Woe's efforts to clarify the evolution and diversity of microbes provided an invaluable service to ecologists and conservationists. Berge's Manual of Systematic Bacteriology is the main resource for determining the identity of bacteria species, utilizing every characterizing aspect. Berge's manual was first published in 1923, and the current volumes differ drastically from previous volumes in that many higher taxa are not defined in terms of phenotype, but solely on the sequence information from 16S ribosomal RNA phylogeny. As a practical matter, phenotype is used to identify microbes in the absence of sophisticated sequencing analysis. Cell size, shape, and arrangement are visualized immediately with the microscope. Distinctions between eukaryotic and prokaryotic cells are obviated. Staining characteristics can reveal structural differences, which include the presence of capsules, flagella, and endospores. Morphological arrangements of cells, such as chains, tetrads, or sarcina, can be visualized. Intracellular localizations may be seen. And of course, the gram and acid-fast stains can help determine the direction of antibiotic therapy. Recognizing different bacterial growth morphologies on agar plates and other media is a useful and often crucial step in the identification process. The growth of bacteria on different media types may provide information which includes the colony features, descriptions of size, shape of colonies, surface characteristics, consistency, optical properties, and color. Differential media contain indicators that expose further differences between organisms. Media can be formulated in such a way that differences in the biochemistry between organisms will be detected. Visible or color changes take place either during incubation of or after the addition of reagents. The visible or color changes reveal the presence or absence of chemical action and thus produce a positive or negative result. Table 10.5 from your textbook lists some of the key biochemical tests performed to identify bacteria. A dichotomous key is a tool that allows the user to determine the identity of items in the natural world, such as trees, wildflowers, mammals, reptiles, rocks, and fish. Dichotomous means divided into two parts. Therefore, dichotomous keys always give two choices in each step. For bacterial identification, the keys consist of a series of choices, selected by the positive or negative results of a series of biochemical tests. As metabolic characteristics are determined, the use of the dichotomous key for bacteria may lead to the identification of the bacterial strain. The metabolic tests may be carried out individually or with commercial multiple test systems. These systems contain individual chambers which can be simultaneously inoculated, then incubated, and then interpreted. Molecules on the surface of bacterial cells can be used as identifying markers. They may be detected by marker-specific antibodies. Identifiable markers may include bacterial surface proteins, surface carbohydrates, and surface lipids. Specific structures whose proteins or molecules can be recognized with antibodies include structures like pili, flagella, fimbriae, and bacterial capsules. Every microorganism has its specific fatty acids in their plasma membranes. Analysis of these fatty acids, this is referred to as a FAME profile or microbial fingerprinting, may be used as a tool for microbial identification. To perform FAME analysis, a bacterial culture is taken and the fatty acids are extracted and chemically converted to methyl esters. The volatile derivatives are then introduced into a gas chromatograph, and the pattern of the peaks help to identify the organism. This is widely used in characterizing new species of bacteria and is useful for identifying pathogenic strains. This slide contains a question to reflect upon and use the presented information.